Welcome to this inspiring journey into the world of nanostructures for biosensing applications. Today we're going to explore how structures smaller than a human hair are revolutionizing medical diagnostics and could save millions of lives. Let's start with the fundamentals. What exactly are nanostructures? Nanostructures are materials with at least one dimension between 1 and 100 nanometers. To put this in perspective, a nanometer is 1/1000th of a meter. 1 nanometer equals 10 to the negative 9 meters. A typical human hair is about 80,000 nanometers wide, so we're talking about structures that are hundreds of thousands of times smaller than a human hair. At the nanoscale, we're working at the same size as biological molecules. DNA is about 2 nanometers wide, proteins are around 5 nanometers, viruses are about 100 nanometers, and bacteria are around 1,000 nanometers. Now you might ask, why are nanostructures so special for biosensing? What makes them different from regular sensors? The answer lies in physics. At the nanoscale, the surface area to volume ratio increases dramatically. This means more surface area is available for biological molecules to interact with. For a sphere, the surface to volume ratio is 3/r. As r gets smaller, this ratio gets larger. This is inverse proportional to size. Let's do a quick calculation. Compare a 1 micrometer sphere to a 10 nanometer sphere. For the 1 micrometer sphere, which is 1,000 nanometers, the ratio is 3 divided by 1,000, giving us 0.003 per nanometer. For the 10 nanometer sphere, we get 3 divided by 10, which is 0.3 per nanometer. That's 100 times larger. The nanoscale structure has 100 times more surface area per unit volume. This means 100 times more binding sites for target molecules, leading to dramatically improved sensitivity. Now let's dive into one of the most powerful phenomena in nanobiosensing, surface plasmon resonance, or SPR. First, what is a surface plasmon? It's a collective oscillation of electrons at a metal dielectric interface. Imagine a gold nanoparticle. When light hits it at the right frequency, the free electrons oscillate collectively back and forth. This creates an intense electromagnetic field at the surface. The resonance frequency depends on the size, shape, and local dielectric environment of the nanoparticle. This is the key to biosensing. The maximum wavelength is approximately equal to lambda p times the square root of 1 plus 2 epsilon m, where epsilon m is the dielectric constant of the surrounding medium. When target biomolecules bind to the nanoparticle surface, they change the local dielectric constant. This shifts the plasmon resonance wavelength, which we can measure optically. The wavelength shift delta lambda equals m times delta n times the quantity 1 minus e to the negative 2 d over ld, where m is the sensitivity factor, delta n is the refractive index change, d is the adsorbate thickness, and ld is the decay length. Typical sensitivities for gold nanoparticles range from 100 to 500 nanometers shift per refractive index unit. This allows detection of incredibly small amounts of biomolecules. Let's work through a real example. Glucose sensing for diabetes management. Normal blood glucose levels range from 70 to 100 milligrams per deciliter. Diabetics need to monitor this constantly. Let's convert this to molar concentration. 100 milligrams per deciliter equals 1 milligram per milliliter, which is approximately 5.5 millimolar, using glucose molecular weight of 180 grams per mole. The challenge is to detect 5 millimolar glucose selectively in a complex mixture like blood, which contains thousands of other molecules. We use gold nanoparticles functionalized with glucose oxidase enzyme. This enzyme specifically binds glucose and catalyzes its oxidation. Glucose plus oxygen, in the presence of glucose oxidase, produces gluconic acid and hydrogen peroxide. The hydrogen peroxide produced can be detected electrochemically or optically. The current or optical signal is directly proportional to glucose concentration. The electrochemical current I equals NFAD times C over L, where N is the number of electrons, F is Faraday's constant, A is electrode area, D is diffusion coefficient, C is concentration, and L is diffusion layer thickness. Now let's explore another fascinating class of nanostructures, quantum dots. These are semiconductor nanocrystals with remarkable optical properties. What makes quantum dots so special? They exhibit quantum confinement effects. When a semiconductor crystal becomes smaller than about 10 nanometers, comparable to the exciton Bohr radius, quantum mechanics takes over. The bandgap energy Eg equals the bulk bandgap plus h bar squared pi squared over 2 mu r squared, where mu is the reduced mass and r is the particle radius. As the quantum dot gets smaller, the bandgap increases, and the emission wavelength shifts toward blue. This is the opposite of what classical physics would predict. By precisely controlling the size during synthesis, we can tune the emission color across the entire visible spectrum. A 2 nanometer cadmium selenide quantum dot emits red light at 650 nanometers, while a 5 nanometer dot emits blue at 500 nanometers. This size tunable emission makes quantum dots perfect for multiplex biosensing, where we detect multiple targets simultaneously. We can attach red quantum dots to antibodies for protein A, green dots to antibodies for protein B, and blue dots to antibodies for protein C. When we add a sample, each antibody quantum dot conjugate binds to its specific target. We can then use a spectrometer to detect all 3 colors simultaneously, telling us the concentration of all 3 proteins at once. A clinically important example is the cardiac biomarker panel for heart attack diagnosis. We need to detect troponin I, creatine kinase MB, and myoglobin simultaneously. Traditional methods require separate tests and hours of waiting. With quantum dot multiplexing, we can get results in 15 minutes from a single drop of blood. This speed can be the difference between life and death in emergency departments. Let's explore another revolutionary nanostructure, carbon nanotubes. These are rolled-up sheets of graphene with extraordinary properties. Carbon nanotubes are hollow cylinders made of carbon atoms arranged in a hexagonal lattice. They can be single-walled or multi-walled. A single-walled carbon nanotube has a diameter of just 1 to 2 nanometers, but can be micrometers long. This extreme aspect ratio gives them unique properties. Carbon nanotubes have exceptional electrical conductivity, around 1 million Siemens per meter. This is better than most metals. They also have extremely high surface area, between 1,000 and 3,000 square meters per gram. That's like having a tennis court worth of surface area in just 1 gram of material. A powerful application is dopamine detection for Parkinson's disease monitoring. Parkinson's disease is caused by dopamine deficiency in the brain. Being able to measure dopamine levels accurately could revolutionize treatment. Normal extracellular dopamine concentrations range from 10 to the -7 to 10 to the -6 molar. That's incredibly low, requiring highly sensitive sensors. Carbon nanotube electrodes modified with specific polymers can detect dopamine at nanomolar concentrations, well below physiological levels. Dopamine undergoes a 2-electron oxidation to dopamine oquinone plus 2 protons and 2 electrons. These electrons create a measurable current. The peak current IP equals 2.69 times 10 to the 5th times n to the 3/2 times A times D to the 1/2 times C times V to the 1/2, where n is electron number, A is electrode area, D is diffusion coefficient, C is concentration, and V is scan rate. With optimized carbon nanotube electrodes, we achieve a limit of detection of 10 nanomolar with a signal-to-noise ratio of 3. This sensitivity opens doors for real-time brain chemistry monitoring. Now let's discuss the theoretical foundation of biosensing, the Scatcherd equation, which describes binding interactions. The Scatcherd equation is R over C equals NK minus RK, where R is moles of ligand bound per mole of receptor, C is free ligand concentration, N is number of binding sites, and K is the association constant. This equation describes how target molecules bind to the sensor surface. It's fundamental to understanding sensor response. From the Scatcherd equation, we can derive the Langmuir isotherm, which describes surface coverage. Theta equals Kc over 1 plus Kc, where theta is the fractional surface coverage, K is the equilibrium constant, and C is the analyte concentration. At low concentrations, where Kc is much less than 1, we get approximately theta equals Kc, a linear response. At high concentrations, where Kc is much greater than 1, theta approaches 1, meaning all binding sites are occupied, and the sensor saturates. The dissociation constant Kd, which is 1 over K, defines the sensor sensitivity. Lower Kd means tighter binding and better sensitivity. Kd equals Koff over Kon, where Koff is the dissociation rate constant and Kon is the association rate constant. For antibody-antigen binding, typical Kd values range from nanomolar to picomolar, indicating very tight binding. A Kd of 10 to the negative 9 molar means the sensor reaches half saturation at 1 nanomolar concentration. This defines the optimal working range. One of the most exciting aspects of nanobiosensors is signal amplification. How do we detect even lower concentrations? Strategy 1 is enzymatic amplification. Each enzyme molecule catalyzes thousands of reactions per second. Typical enzyme turnover numbers range from 1,000 to 1 million per second. This means one enzyme molecule can generate millions of detectable product molecules. For example, horseradish peroxidase is commonly used as a label. It catalyzes the oxidation of chromogenic substrates, producing colored products. With enzymatic amplification, we can achieve amplification factors of 1 million to 1 billion, dramatically improving detection limits. Strategy 2 uses nanoparticle labels. A single binding event can be tagged with a nanoparticle containing thousands of reporter molecules. For instance, a 50 nanometer gold nanoparticle can carry 100 to 1,000 fluorophore molecules on its surface. Each binding event thus generates a much larger signal. Let's examine a real-world application that impacted billions of people: COVID-19 detection using nanobiosensors. Traditional PCR tests require sample transport to centralized labs and take 1 to 3 days for results. During a pandemic, this delay is catastrophic. Nanobiosensor-based rapid antigen tests provide results in 15 to 30 minutes at the point of care. This speed enables rapid isolation and contact tracing. The test uses gold nanoparticles conjugated to anti-SARS-CoV-2 antibodies. When viral proteins are present, they bind to the antibodies and the gold nanoparticles accumulate at the test line, creating a visible red color. These tests can detect 10,000 to 100,000 viral copies per milliliter. While less sensitive than PCR, they're sufficient for detecting infectious individuals. The key insight is that infectious individuals typically have viral loads of 1 million to 1 billion copies per milliliter, well above the detection limit. Let's look toward the future. Where are nanobiosensors taking us? Continuous glucose monitors using nanostructured electrodes are already transforming diabetes care. These devices measure glucose every few minutes, 24 hours a day. The next frontier is continuous multi-analyte monitoring. Imagine a small patch that continuously measures glucose, lactate, cortisol, and inflammatory markers. Such a device would provide real-time health monitoring, alerting you before problems become serious. This is personalized medicine at its finest. Another exciting direction is early cancer detection from circulating tumor cells. Nanostructured chips can capture and analyze these rare cells from a simple blood draw. Cancer patients have just 1 to 10 circulating tumor cells per milliliter of blood among billions of normal cells. This is like finding a few needles in a massive haystack. Nanostructured surfaces with antibody coatings and optimized topography provide the sensitivity and selectivity to capture these rare cells, enabling early detection when treatment is most effective. Now let's be scientifically rigorous. What are the fundamental limits of nanobiosensors? What prevents us from detecting a single molecule every time? First, thermal noise is unavoidable. At room temperature, molecules are constantly in random motion. The thermal energy equals 1/2 kBt, which is 2.07 times 10 to the negative 21 joules at 300 Kelvin. This thermal energy causes fluctuations in any measurement. Molecules randomly bind and unbind from the sensor surface even in the absence of target. Second, we have shot noise from discrete molecular events. When counting individual molecules or electrons, we get statistical fluctuations. The signal-to-noise ratio equals n over square root of n, which simplifies to square root of n. To improve SNR by a factor of 10, we need 100 times more molecules. In practice, nonspecific binding is often the limiting factor. Other molecules in the sample can stick to the sensor surface, generating false signals. Minimizing nonspecific binding requires careful surface chemistry, blocking agents, and washing steps. This is as much art as science. Let's talk about a crucial question. How do we make these advanced technologies accessible to everyone, not just wealthy nations? Traditional laboratory tests cost $50 to $500 per test in developed countries and are often unavailable in developing regions. The goal for nanobiosensor point-of-care tests is to bring the cost below $1 per test. This requires mass production and simple designs. Paper-based nanobiosensors are showing great promise. By printing nanoparticles on paper substrates, we can create tests that cost just pennies to manufacture. A paper card with 3 detection zones, each containing different functionalized nanoparticles, can simultaneously test for multiple diseases. No electricity, no refrigeration, no trained personnel required. This technology could revolutionize healthcare in resource-limited settings, bringing advanced diagnostics to rural clinics and remote villages worldwide. As we develop these powerful technologies, we must consider the ethical implications. Continuous health monitoring raises privacy concerns. Who owns your health data? Who can access it? These are not just technical questions, but societal ones. We must ensure that these technologies don't widen the gap between rich and poor. Innovation must be coupled with strategies for equitable access. Rapid deployment faces regulatory challenges. How do we ensure safety and efficacy while not stifling innovation? The COVID-19 pandemic highlighted both the need for speed and the importance of rigorous validation. Let me summarize why nanostructures are revolutionizing biosensing. The massive surface-to-volume ratio at the nanoscale enables sensitivities orders of magnitude better than bulk materials. Quantum confinement effects provide unique optical and electrical properties that we can tune precisely by controlling size and shape. We can integrate multiple detection modalities, optical, electrical, and mechanical, into a single nanoscale platform for comprehensive analysis. These advances enable true point-of-care testing, bringing laboratory-quality diagnostics to the patient's bedside, the doctor's office, or even the patient's home. Looking ahead, the future lies in integration and intelligence. Artificial intelligence algorithms can analyze complex patterns from multi-analyte sensors, detecting disease signatures that humans might miss. Integration with smartphones and wearables will make health monitoring seamless, with results transmitted instantly to your doctor or AI health assistant. Implantable nanobiosensors could continuously monitor chronic conditions like heart failure or diabetes, automatically adjusting medication delivery in real time. Imagine this integrated system. Nanobiosensors measure your biomarkers, transmit data to your smartphone, which sends it to AI systems in the cloud that analyze trends and alert your physician if intervention is needed. This is the future of healthcare. If you're inspired by this technology, what can you do? How can you contribute to this revolution? This field requires true interdisciplinary collaboration. Chemists, physicists, engineers, biologists, and clinicians must work together. Key focus areas include advanced materials, science for new nanostructures, surface chemistry for selective binding, and data analysis for extracting meaningful information from complex signals. Clinical validation is crucial but often overlooked. We need researchers dedicated to testing these devices in real clinical settings with actual patients. Let me conclude with a vision of where this technology can take us. We can transition from reactive healthcare, treating diseases after symptoms appear, to predictive healthcare, detecting and preventing diseases before they cause harm. Nanobiosensors promise the democratization of advanced diagnostics, making cutting-edge medical testing available to everyone, everywhere. Early detection enabled by sensitive, affordable nanobiosensors can dramatically reduce healthcare costs by catching diseases when they're most treatable. The cost of prevention is much, much less than the cost of treatment. This simple inequality drives the entire field of early diagnostics. Ultimately, this technology has the potential to save millions of lives annually through earlier diagnosis of cancer, infectious diseases, cardiovascular conditions, and countless other ailments. Let me leave you with this final thought. The most sophisticated nanofabrication, the most elegant equations, the most sensitive detectors, all of this is meaningless unless it serves humanity. Technology must be guided by compassion and ethics. As you pursue your research and careers, always ask, who benefits from this? How can we ensure equitable access? Are we making the world better? You are the generation that will transform healthcare using nanotechnology. The challenges are immense, but so is the potential. Go forth and make a difference. Thank you.